So we'll now hear from Jordan Miller. Jordan is a postdoctoral fellow at the University of Pennsylvania, a founding member of the Hive 76 in Philadelphia, and a RepRap core developer. Welcome, Jordan.
All right, can everybody hear me? All right, yeah, thanks Dustin, thanks Katarina, thanks to all the organizers. I'm really excited to be here to tell you about our work using a RepRap 3D printer to do basic research in regenerative medicine. So let's start off, what is regenerative medicine? So there's actually two competing ideas and kind of visions for the future of human medicine. And the first one is kind of depicted here that a lot of people are familiar with from the past several decades of television. It's the bionic man. The idea that basically any problem in the body can be treated with a device. So if you need a total knee replacement, if you need a hip replacement, if you need a pacemaker, if you need an ear implant to be able to hear, they have retinal implants now, whole limb replacements. And these strategies have been very helpful for the people that need them. But what happens is that these devices are not permanent in the body. They need to be adjusted, they need to be revised later. Like a hip implant or knee implant may only last about 20 years, and they don't ever fully integrate with the body. So there's an alternative hypothesis that's been really kind of envisioned in the last couple of years, and it's that of regenerative medicine. And the National Institutes of Health has picked for their mascot for regenerative medicine, Prometheus from Greek mythology. Now Prometheus was chained to a rock and sentenced to have his liver eaten every day. This is not the most optimistic vision for the future of humanity. But what gets the National Institutes of Health excited about this idea is that every day after his liver was eaten, it would naturally regenerate again. So the idea is, can we develop strategies to either induce or help replace failing organs and tissues that can't be treated with any kind of known device nowadays. So tissues like the liver, the pancreas, the kidney, the brain. And can we do this in a way that it will fully integrate with the body and help replace function? So basically the idea of this theory is that cells are always going to outperform devices for implants in the body. So this has already had very successful strategies in the clinic. So these are already in patients, helping to heal them from, so this is a 2D scaffold seated with the patient's own cells that can be used as a skin replacement after a burn. There are corneal implants that are being used. There's an artificial bladder that's made. They take a 2D scaffold, they can sew it up into a 3D shape, put the patient's own cells on there, and that makes a functioning bladder. The challenge is really, how do we translate these types of ideas into 3D structures? So these 3D organs, like I showed before, so the liver and the kidney. And if you notice, all of these are thin tissues, and they don't have a blood supply. And this is really the challenge. We want to look at this challenge. If we look at the anatomy of the liver, there's a perfect example here. So the liver is a very large organ, but almost half of the volume of the liver is blood vasculature. And the vasculature has a very complicated, interconnected 3D multiscale structure. So multiscale means it has many different sizes of diameters of channels all connected together. And we really have no idea how to make structures like this. I know a lot of people that are 3D printing in the audience may say, I'm going to print that, and we'll talk about that in a minute. But we have no idea really how to make this yet. And so this is one approach that we think may help to address this space over the long term. So one thing you may think, those of you familiar with 3D printing, we've seen some demos of that today, maybe we can just print the cells and the gel, so the supporting matrix. It's mechanically, it's something like Jello. Can we print that in 3D, encase the cells, make everything except for the vasculature, and then pump all the blood through and keep the cells alive? So this has worked for some types of cells, but there are certain cell types, like the liver cells, for example. These are very interesting from a therapeutic perspective for treating a patient, but they're actually very fragile cells, and they don't survive this process very well. And we don't really know how to change the cells to make them survive this process yet. This is a very active area of research. But we were kind of unsatisfied with these types of technologies, and we decided to try to figure out, is there a different way that we could do this? So if you take a step back, sometimes you can get insights by kind of reconsidering everything from the beginning. So this is from the Body Worlds exhibit. I don't know if any of you have ever seen it, but this is a whole organ vascular cast of a human organ. I think it's a kidney. And what you just see here is this really complex architecture. And when I first saw this, immediately I was thinking, well, this is really what we're trying to make, this vasculature. What if we can make something in this type of shape first? And then maybe we could put these fragile cells, these cells don't survive 3D printing, put them around there afterwards, and then maybe remove this material, and then we would have our vasculature. So it's kind of a weird idea. How would we do that? We didn't really know for a while. So at the time, that was kind of kicking around in my head. I'd seen a bunch of 3D prints that had come off, and this is kind of interesting here, this kind of structure. And these kind of materials, you could theoretically put cells around them. They can be made biocompatible, but you can't remove them afterwards. Anything that you would use to dissolve these materials, this is basically an epoxy resin. These are these bioplastics. It would just kill your cells immediately. So luckily, I had dessert afterwards, and you should always have dessert. So when I had dessert, you actually notice here, not the flan, but look at this sugar cage here. So this sugar cage, this 3D print, and this organ vasculature, if you really want to think about it, it's really just a series of tubes. So what we're trying to do now is take this sugar type of structure. It's a very strong material. We know it's biocompatible. It's in your bloodstream already. Can we figure out a way to 3D print this sugar, something with a 3D printer, something that is accessible to us, make something that basically functions like a vasculature to try to keep these cells alive? So that's what we set out to do. This is kind of the schematic overview. So we're trying to 3D print something like a filament network, something made out of the sugar. We're going to pour this sort of gel containing the cells all around the sugar. The gel will solidify and trap the cells. Then we can dissolve the sugar, and wherever the sugar was, that has the vascular architecture that we want. We can then perfuse it with blood and try to keep cells alive. So how are we going to do this? Well, if you look at some 3D printers that are out there, this is kind of the scenario that you go through. So you call up these companies, say, hey, I want to spend $100,000 to buy your printer, and I just need the schematics because I want to make it print sugar for biocompatibility studies. And they just don't even know what to say to that. So you have no ability to really teach companies what they could even do with their own technology. They haven't released a source code. This is really just completely backwards. So you have no ability to instruct and to modify these printers. And it doesn't matter how much money you have, you're not going to really be able to set up this type of a relationship with these types of companies. So I just want to say also that we're here for open hardware. Science fundamentally is about open. So open source is the reason why science works. So if you do an experiment and you tell me about it, if I can't repeat that experiment, then actually you're in trouble, not me. So you haven't explained it correctly enough for me to repeat it and to verify your results. The great thing is also, from our perspective doing science, is that open source, it makes it trivial to get collaborators. So anyone in the RepRap group, you can just get on IRC and say, hey, that board you made is really awesome. Can you add another MOSFET for me because I want to do this other thing? Like, oh yeah, sure, I'll ship it out next week. It's fine. And the reason that's true is because when you release the source code, that necessarily is creating open standards that other people will follow. You've released it, it's under Git revision, and so you know exactly what those files are. And I would just say, there's nothing wrong with going closed source per se, but I think that what people should consider if they're trying to think about going closed source, or if they're starting out closed source, really it may not affect you right in the immediate time. But if you think about it, you're closing off the future of what your company could be because you can't really envision everything that your device would do. So I guess I'll kind of running out of time, sorry. But so this is how it works. This is now we're using a RepRap 3D printer. We've modified the design, made the extruder able to extrude this molten sugar. This is how it works. We're building up this wall. This extruder design that we made, we modified it, we put it all back on Thingiverse, it's on GitHub. You can download it and try it today. These are edible sugars. This is me pouring a gel to encase that sugar lattice in there. And this is how the process works. So you can make really interesting structures. The sugar, mechanically, it's a glass. So anything you can make from 3D glasswork, you can make essentially with this 3D printer. Then we're going to dissolve all the sugar away. We can now pump actually human blood through these channel architectures to be able to sustain the cells that we're going to encase there. And we just have an example here. So there are cells that line all of your blood vessels. And what we're doing here is we're taking those cells, we label them in red, and we're injecting them into these channel architectures that we've made. We can make them line all the channels. So we have this debate, how much of that vasculature do we need to make to be able to keep cells alive? And so we went sort of, this is about 500 micron, this is about 200 micron. And when you put these together over time, now we're looking down one of those 200 micron channels that you saw here. This is a channel that we made, and the cells are starting to make capillary sprouts coming off of all of these different vessels that we patterned from the sugar-based channels. So you can see that very clearly here, and this is kind of just an image of that. So I'm really pleased to tell you we got this published in a peer-reviewed journal article, and I think we have a really great team in the lab at Penn and at MIT that we were working with. But I think this is a really big win for the whole open source community. We were able to put them all in the acknowledgements. So you can see all the different open source projects that we really utilized and we really relied on to get this stuff done. And I really encourage you guys to check out those projects. And I would just kind of leave you with the idea that really what science needs from you is we need open technology platforms and not appliances. And so when you think about releasing stuff, when you think about releasing stuff, you really want to think about the relationship you want to have with your consumers, the relationship you want to have with your community, and how to get that going forward. I guess just really briefly, kind of the future where we think this could go, kind of another vision for this, is kind of taking that vascular architecture I showed in the beginning from the liver. You can extract that, make an STL file that you can 3D print. You can also do network-based analysis of this to reconstitute something entirely synthetically on the computer. This is, of course, using another open source project called Blender. They have a really nice tree renderer in there based on parametric design files. You can then make that into your G-code file. You can 3D print that, and then theoretically this would be in something like sugar, and then you'd basically encase that in there. So I guess we had one more application, which is now we're using this extruder because it's open source for fun. We're using this in our hackerspace in Philadelphia at Hive 76. So here we're actually extruding molten chocolate. So this is not chocolate frosting. This is solid tempered chocolate. And you can see that it's solidified down here and it's molten here. So I'm interested in talking with people about the idea of maybe using this as a way to fund some of the research that we're doing, whereby the actual technology needed to adjust it so we can 3D print chocolate better would directly impact the technology used to 3D print the sugar for the regenerative medicine research. So thank you.